Miss Litton, and this is AP Bio last review before your next exam, which you are taking with a partner. Why didn't you say friend? I thought you'd say friend. Okay, so this was the latest list of questions that people had, and so I'll try to review these big ticket items. And these came out of after what I talked to you today about in class about what I would pay attention to. So I will just kind of work my way through them, and if there's something else on there that you want to ask me about at the end, I'm happy to answer that unless I have to look. Okay? All right. Oh, say hi. Hi. All right, here we go. So the first one, I'm probably not going to come back to this list again. I'm just going to work, because I tried to type up the list on slides to meet what you said to me today. So the first one is gene regulation in cancer. So if I ask you a question about, you need to be very careful when I ask you a question. I might ask you about things that regulate the cell cycle normally that are proteins or something like that. And if I say something that regulates the cell cycle normally, I'm not talking about these genes right here related to cancer. What am I talking about? Cyclins and what else? Checkpoints. Exactly. I'm talking about cyclins and checkpoints. So I just want to address that. Remember, cyclins have to build up different cyclins at different times in the cell cycle. You have to pass through checkpoints. Okay? Those are signals to trigger the cell. Yes, you may move forward in the cell cycle. Keep in mind the checkpoints are just making sure what you've done so far is correct, right? So I have to verify that it's correct, and I have to have a buildup of a cyclin in order to move into the next step. Are you all right with that? Then there are genes, okay? Those genes that we have, like a proto-oncogene could code, if I say specifically, tell me about two genes, a proto-oncogene could code for a cyclin, right? Because proto-oncogenes are in a position of what? Power. And they control the cell cycle by facilitating the cell cycle. They're part of the cheering squad that says, move forward into the next stage. Whereas tumor suppressor genes are not about facilitating the cell cycle, they're about what? Stopping or inhibiting the cell cycle. Okay? And it's a cell cycle, it's not like you have to use tumor suppressor genes all the time, right? What do we have more of? Proto-oncogenes and tumor suppressor. Tumor suppressor come into play when a proto-oncogene has mutated into a oncogene, right? Proto-oncogenes you need all the time. There's nothing wrong with them. They're perfectly perfect. The only problem is when a proto-oncogene in a position of power goes bad and becomes and mutates into an oncogene, then all of a sudden it's telling you to go forward when you shouldn't go forward in the cell cycle, okay? But they're a normal part. Proto-oncogene, normal. Mutated proto-oncogene, oncogene, bad, okay? And that's a cancer-causing gene, and it causes those cells to proliferate. What was our classic example of a gene that has gone bad? RAS, because it's like stepping on the gas. Normally, RAS would be triggered by a receptor, and the receptor would have been triggered by some sort of what? Growth factor, a chemical of some sort, and that receptor would then trigger RAS. But in this case, RAS is just saying, the light is green, and he is moving forward and acting like a signal has been received. So this is an example of not good communication, but bad, poor communication, right? I didn't say to go, okay? But he is saying yes and triggering the DNA, and that gene, when it gets expressed, maybe it is making a cyclin when it shouldn't, okay? So that's what a bad, if the RAS was working fine, it would just be called a what? What coded for it? Yeah, a proto-oncogene was coding for a good RAS. An oncogene codes for a what? Bad RAS. Okay, but it's a normal part. Now, tumor suppressor genes are when you have a problem and it's out of control. These genes code for proteins to help regulate the cell cycle. And what's our classic gene there? P53. Good. And what checkpoint is it in? Yeah, G1. And P53 in the G1 checkpoint says, you know what? We might have had a problem. Let's stop. Let's see if we can fix our DNA. So it will literally turn on genes that would make proteins that would A, stop it from moving forward in the cell cycle, correct anything you could possibly correct. If you can't correct it, then you say do what? Apoptosis. And what is it like to use for that? Caspases. Good. Okay? So those are those two genes. Do you have any questions you would like to ask me about those? ¿Dónde? ¿Cómo está? Yeah, I don't know what I'm saying. All right. Next thing on the list, is cell signaling. Okay, like in the immune system. All right. So interferons versus interleukins. So when you talk about interferon, okay, you interfere. You're trying to interfere with virus life cycle. So remember we talked about this with plant cell signaling today, right? When we talked about when plants have a pathogen in them, it triggers an alarm throughout the whole plant to say, hey, be careful, there's viruses here, some sort of pathogen. That's the same thing that happens here. In an interferon, you've got some sort of virus. This is activating genes that are releasing a chemical. So this is how one cell warns another cell, protect yourself. Okay? And the way it tells it to protect itself is when that gets released, that interferon binds to a receptor. This, look, signal transduction, you've seen it before, but this time it makes a particle or a protein that will destroy the virus parts as soon as it invades. Now, you wouldn't know to have those ready unless you had been warned, right? You've been warned, so you're ready to go. As soon as that virus comes in, you're like, I'm done with you. And you never let it gain a foothold in that particular cell so it could make more of itself. Okay, so that is your interferon. Interleukin, I have a little chart here for you. I might have put this in your notes, I can't remember. Cytokines could be several different things because you had asked me to add that to the list, right? So cytokines could be several different things. These are just the way one cell can talk to another cell in your immune response. Specifically, interleukins are things by white blood cells that are amplifying the immune response. They're making the macrophages eat more. They're making the B cells release more antibodies. They're making cytotoxic T cells kill more. It's the Red Bull of the immune system, and it amps it all up. So interleukins. Okay. Ask your bio buddy. Got any questions? Cool. Does that answer your question? Yes. All right. Next on the list was antibody versus cell-mediated immune response. Now, is this part of your innate immunity or your acquired immunity? Acquired. This is the part that's unique to all of you in the room, right? Because unless you were sitting next to your identical twin that was exposed to all the same things as you, just a second. Okay, yeah. Okay, so unless you were exposed to an identical twin always throughout your entire life, exposed to the exact same antigens, this is going to be unique to you, yes? And so this type of immunity, your adaptive or acquired immunity, this is where you are making fighters, specifically those that originate from B cells and those that originate from T cells, to attack only one kind of pathogen, a single focus. And B cells, their target is the actual pathogen, the virus, the bacterium that has invaded your body. And the way they do that is B cells have receptors, BCRs, B cell receptors, for the foam sparkle virus, okay? And once it has been triggered, I'm a virgin B cell, I would not recognize the dragon foam, I would only recognize sparkle foam, okay? A different virgin B cell would recognize dragon foam. I recognize the sparkle foam antigen. I will then grow and divide, and I will make two kinds of cells. What are the two kinds of cells I will make? Plasma cells. What will plasma cells do? Secrete antibodies, which will mark, which will mark the sparkle foam only for destruction. It will not trigger, the antibodies will not bind to the dragon foam. You see the difference there? So That's wiping out. Those are the free-floating phones and viruses in your body. That's how they handle it. Do the antibodies themselves destroy the virus or the bacterium? No. Now, it could get in the way of it binding to a cell, like a virus binding to one of your cells and gaining access. It can neutralize it. So it's just out there like, oh, I'm a bad guy and I'm marked. Okay, somebody come eat me. Okay? So it may neutralize so it can't get into your cells, but it marks it for destruction. There are like 5 different classes of antibodies, different ways you can be destroyed. Whereas cell-mediated is just like what it says inside of it. It's a cell, and it works. Those cells are T cells, and their targets are your own body cells that have been compromised. Your own body cells that have been compromised. And they are displaying a piece of what compromised them on the surface of their cells. And those cells that could be killed by a T cell are going to display that antigen on the what? MHC1, and that means that you are what? Done. You're going to be killed when you display on an MHC1. However, the amazing thing about your adaptive immunity is it doesn't kill all cells that are displaying that antigen. Who are some cells it would help and not kill? Antigen-presenting cells. Can you give me an example of an antigen-presenting cell? A dendritic cell, a macrophage, a B cell. Okay? These have not MHC1s. They display an antigen on an MHC2. And so when the T cell connects, it knows if it's killing you, if it's killing that cell, an antigen must be getting displayed on its MHC2. If you're killing it, it's on an MHC1. You're what? Done. I'm just going to add this. I didn't have it on the screen, but the T cell stabilizes with an MHC1. It stables it with a protein called CD8. And I always think, oh, it's going to get 8. Okay? But if it binds with a CD2, it stabilizes with, if it displays an antigen on an MHC2, the T cell stabilizes the bonding, the connection, with a protein called a CD4. I am for you, not against you. So there's more machinations than just MHC1 and MHC2. From the T cell come these like arms, these binding arms. And if you get bound with a binding arm 8, which would only fit onto an MHC1, then you're going to get 8. You're done. But if you get bound to that T cell with a CD4 with an MHC2 antigen, then I'm for you, not against it. I won't kill you. Yes?
So you said the B cell just marks the pathogen?
B cell doesn't mark it. Who marks it? Antibody. Who secreted the antibody? Plasma cell, which is a type of specific B cell. Okay, go ahead and now ask your question.
But then which one actually kills the pathogen?
Oh, okay. A macrophage could come and eat it.
Oh.
A cytotoxic T cell. So here's the beauty of this. Remember, you can still have your innate immune response assisting your Navy SEALs. The police officers are still assisting the Navy SEALs. But now there's literally no thinking involved. If you're tagged with an antibody, you're out. You're done. Anything can kill you. Anything that's weaponized can kill you. You can get eaten. You can get destroyed. Any type of killing cell can do that for you. You can enlist your entire army to do that. Now, whereas the B cells, you had just plasma cells, and what was the second cell? Memory cells for next time. T cells have, what are those cells? You tell me. Cytotoxic T cells, which kill, cell-to-cell combat. What else? Helper T cells, which, what's something they could release that would help the immune response? Something, what? What kind of cytokine? Interleukin, right? Okay. They're going to amp up the entire immune response by secreting those interleukins and saying, let's fight the good fight. Does that make sense to you? Okay. What's the third kind of cell? Memory. Okay. And okay. And this was just, I'm going to come back to those memory cells in a minute. This is just another diagram I found where it's showing you in the humoral immune response, it's the actual virus, the bacteria that's triggering the virgin B cells, and those will mature into plasma cells, which secrete antibodies. Those antibodies will mark it, and then you can see, here, let me make this bigger. So you can see those antibodies are marking them for destruction, and then here you can have some sort of macrophage come and eat them or destroy them. Whereas your T cells, okay, you are displaying it. Either it's an antigen presenting cell, displaying on it that because it killed it, and it's like cutting off its head and showing it to everyone on its MHC2. I will help you stabilize by a CD4. Or you nuggets, yeah. And so then it will assist or it will kill if you're displaying on an MHC1. Look at all those cytokines to amp it up. There are cytokine receptors, right? Because if you're going to secrete an interleukin or interferon, you have to have a what? Receptor for an interleukin or an interferon. That's why this is underneath cell signaling. I was making an umbrella, if you didn't know. Okay. All right. Then this was just another one. I was just kind of a summary slide of, everything that is there. And you know what? This presentation that I just made, if this is helpful to you, I can PDF this and put this in the classroom, the stream? Yes. Okay, I will do that. So this, but these are these ones that, anyway, yeah. Okay, so that's just another review of that. I'm going to move on. Okay, next on the list. The difference between centrosomes and centrioles. Okay, centrioles are made out of 9 sets of 3 what? Microtubules. You find them in animal cells. They're perpendicular to each other. They literally have a name, mother-daughter centriole. Okay, you don't need to know all that, but that is what it is. And the centrioles sit inside of a bubble that you would call a centrosome. So centrioles live inside the centrosome. Emanating out of the centrosome will be more microtubules, and this is part of your whole spindle apparatus that's governing mitosis. Okay? So fill in the blanks. Centrioles live inside of centrosomes. Coming out of the centrosomes are microtubules, which form the spindle. Do all cells have centrioles? No. Do all cells have centrosomes? Yes. Do all cells have spindles? Yes. Okay? There. All right. And then I found this picture for you. The Centrosome Cycle. Okay? I just in case you were curious, here you can see the mother-daughter, the centriole engagement. And look right here. The mother-daughter, oh, here, let me make it bigger for you. Maybe. Okay, the mother-daughter centrioles duplicate themselves. New centrioles form near their proximal ends. This takes place during what? Yeah. Okay, so why do animal cells have them and not plant cells? Who knows? Okay, and then we can see them forming that spindle apparatus for you to line up your chromosomes. Okay, next on your list. Oh, multiple feedback. Look, I just said look at your hyperdoc. Read that because that was a content-based thing that I would like you to review. I can tell you right now, you want to be, I would focus on positive and negative feedback loops and ecosystem feedback loops. That's where I would focus my time and my energy. Yes?
By ecosystem, do you mean like a plant?
The positive and negative feedback loops that were in your hyperdoc, it was real concrete what you needed to do on that. It gives you an example of a positive feedback loop and a negative feedback loop.
Like the examples.
Mm-hmm. Yeah, I want people to look at that themselves because that's their work they should have done and it should be a resource for them. Okay, but I'm telling you that's what I would review there. Okay, next. I was asked about haploid and diploid. Your body cells are called what kind of cells? And they are all diploid. That is the kind of organism we have. The only cells that are haploid in our body are gametes, sperm and egg. Okay? Now, we haven't learned that yet. It'll be the first thing we learn of the new unit is mitosis, because then we're going to get ready for sex. Okay? And so, okay? So, a diploid, you have 2 copies of all your genetic information, You have a copy from mom and a copy from dad. So this could be homologous pair 1, homologous pair 2, homologous pair 3. These are diploid. Okay? So you have mom, dad, mom, dad, mom, dad. Now here, okay, and when you make sperm and egg, you have to do mitosis. You don't need to worry about that this chapter. But that is the only way you'll get haploid cells. You'll get either the mom or the dad's chromosome 1, either the mom or the dad's chromosome 2, either the mom or dad's chromosome 3, and you will make a haploid cell. Our diploid number is 46. Okay? So how many pairs do we have if we have 46 chromosomes? 23 pairs of chromosomes. So that means either your eggs or your sperm have how many chromosomes in them? 23. So little Mr. Happy sperm has 23 chromosomes, and Happy egg has 23 chromosomes. 23 plus 23 makes 46. And that fertilization reunites the homologous pairs. Okay, ask your Bubba. You got any questions about that one? Okay. Types of blood. These are red blood cells. If they have A antigens on them, they are called type A. If they have B antigens on them, they are, if they have A's and B's, they are AB blood. And if they have no antigens on them, they are O. Now, if you have an additional RH antigen on it, then this right here would be A positive. A positive. Otherwise, it's A negative. Okay? And you can give blood to anybody that you won't make their immune system angry. Okay? If I am A negative blood, if that is my blood type, okay, that means all my red blood cells have A all over them, and I would never make any anti-A antibodies, otherwise it would hurt me. Yeah. Now, we will talk about the genetics behind that, and when we get into our genetics unit, which is next unit. So I'm going to tell you right now, I did not ask a blood question on this exam. Okay? So just like, don't erase it. You'll want this information later, so don't just put a pin in it for later. Okay? But you don't have to worry about that. Okay? Next. The next one on the list was about immunizations. Everything I told you about B and T cells and acquired immunity, that happens when you get vaccinated, but the antigen you're exposed to triggers the same response without actually having anything to harm you, right? Somebody walked in here right now with a gun. Would we respond the same way? We don't know if it's loaded or not loaded, right? We would be horrified, right? Be like, not my students, okay? Right? It elicits the same response. A vaccine is a gun without any bullets. It triggers all the same adaptive immunity without actually making you sick. That way, if you're ever exposed to the real deal, you can immediately respond to it and neutralize them without actually having to get sick. Does that make sense? Okay. I don't know why you had to go there, Miss Litton, with the gun. Okay, next. Immunizations. That's what I just talked about. Okay. And then the next one was the inflammatory response. Your inflammatory response, that's what we all have. What else do we all have? Before we even enlist the inflammatory response, if I'm just trying to keep that, what do we all have to protect it? Skin, hair, oils. Yeah, you have low pH in your stomach. So those are, I would kind of group those underneath just one heading of barriers to entry, right? So we have barriers to entry in our body. But if something crosses any of our barriers, then you have the inflammatory response. So you're going to elicit, if you look right here, here's the wound and the bacteria are coming in. Who is the most prolific of all of our innate immune responders? Neutrophils, exactly. That's what you have the most of. I think it's like 70% of all white blood cells. I'm not 100% sure, but I think it's somewhere around there. Okay? They're on the scene. They are eaters. They're immediately attacking, triaging the situation, sending off alarms. Next can come monocytes because they're smaller, but the monocytes get out and then they enlarge into what? What do the monocytes enlarge into? Macrophages. They last longer and they can eat more. Okay? You have mast cells. Mast cells are triggered by immunoglobulin E2. Okay? If allergies. So if that binds on, okay, if that binds on, then to a mast cell, the mast cell will release what? Histamine. Histamine causes what? Vasodilation. So it causes the space between your endothelium, the walls of your capillaries, to get bigger. So every time your heart beats, just due to hydrostatic pressure, the force in the blood due to the heart, more fluids get out, which can cause swelling. It can also cause pain. It causes that local area to maybe kill off the bacteria. That's all part of the inflammatory response. Okay? So that those fighting cells can get there a little bit easier. Allergies are just an inflammatory response to something that can't actually hurt you, but does, right? It shouldn't hurt. Peanut butter shouldn't hurt you, okay? But some people are very afraid of peanut butter because their inflammatory response is so great, so much swelling, right? So much vasodilation that their blood pressure drops and they can go into what? Yeah, anaphylactic shock. Or so much swelling in that area that it blocks off their trachea so they can't breathe. Okay? So that is just a hyper-inflammatory response. And because once you're exposed to that, then you start to build up, your body has determined, oh, peanut butter is super bad, peanut butter is horrid. Now you also will build up what? Memory cells. You'll respond faster and stronger each time. Faster and stronger each time, which is, then it becomes lethal. Okay? Okay. Other questions that you have? Tomorrow's, if something comes up, like you study tonight, you're like, oh, I wish I would have asked her. Okay? Tomorrow is an open 7th period just for questions. So you can write those questions down, and then you can come and see me tomorrow if you have any other additional questions. You know that I love you. You feeling the love? Okay. You're not going to wait till tomorrow to study, right? When are you going to start? Last week. Yesterday. Yeah. Okay. Wait, I have a question. Wait, pause, I have a question. I can't even hear you. What?
Confirm our entire test isn't partners or just like...
Yeah, the whole thing. But it makes sense. Cell communication. You need to talk. Wait, I got another question. Hey, be respectful. Go.
Okay, so how does negative phototropism occur?
Okay, so negative phototropism, you don't have to worry about the chemistry behind that. No, okay, what else? Okay, make good choices. I love you. The end. Yes.